These are your Chapter 2 notes from your textbook. We need to review a little bit of chemistry, just make sure we're all on the same page. Matter, remember, is anything that takes up mass, or it's anything that has mass and takes up space. Elements can be found on the periodic table. They are found in nature, and they contain one type of atom. So, for example, carbon is an element, and so carbon would only contain carbon atoms. A compound contains two or more different elements. So, for example, a compound would be carbon. A compound would be glucose, which is carbon, hydrogen, and oxygen. An element would be oxygen. It would even be O2, would be an element. A compound would be carbon dioxide, which has carbon and oxygen combined. Essential and trace elements: carbon, hydrogen, oxygen, carbon, hydrogen, nitrogen make up 96% of living matter. These are what we call our essential elements. Calcium, phosphorus, potassium, sulfur, and a few others will account for the other 4%. That's why we call them trace. 96% of life is going to be made up of oxygen, carbon, hydrogen, and nitrogen. A few elements are required only by, not be certain species, by certain species and not all living things. Example, iodine is required by vertebrates for the thyroid gland, but it's not required for other living things. Some elements are toxic to living things. For example, arsenic can be toxic to humans in particular. On page 32, there is a case study of evolution of tolerance of toxic elements. I want you to read through that, and I believe in your notes there is something that you need to write about that. So if you need to pause the video and go ahead and do that, you may. This is just a table showing the elements of the human body, trace versus essential. Review of chemistry. So here you're not going to be tested on the AP exam or really any exam in my class or quiz or anything else over this material, but it's a really good idea to know it and have that background knowledge. And if you have forgotten it from chemistry, I would recommend you go back to your textbook in Chapter 2 and look it up and maybe make some notes to yourself, or at least just read the summary and remind yourself of these things, because where it comes into play is particular when we start talking about enzymes or photosynthesis and cellular respiration. If you don't understand what an electron is, you're going to have a really hard time understanding what the electron transport chain does. Or if you don't know what a proton is, when we use the term proton and H plus interchangeably, you're not going to get that because you have forgotten what a proton even is. So please go back and remind yourself of this material if you have forgotten it. This just is a quick summary. If it kind of jogs your memory, remember that in an atom, the nucleus is the center. It's made up of your neutrons and your protons, and then the electrons are going to orbit the outside, and they can actually bounce around in orbital level orbits, and they can move electron levels. I don't remember if you remember that from chemistry. And then those electrons are what can be shared between different atoms to create a bond. We'll talk about bonds here in a little bit. Isotopes are different atomic forms of the same element. In nature, an element occurs as a mixture of its isotopes. Radioactive isotopes, one in which the nucleus decays spontaneously, giving off particles and energy. When decay leads to a change in the number of protons, it transforms the atom to an atom of a different element, which can be useful to biologists. So when that decay leads to a change in the number of protons, remember protons are found within the nucleus, they have that positive charge, it's going to transform the atom to an atom of a different element. Remember, isotopes are going to be determined by their proton number. I hope some of this may be jogging your memory from chemistry. If I go too fast, feel free to pause the video so you can write in your notes. I'm trying to go a little slower, keeping that in mind. So what are the radioactive isotopes' uses for biologists? Well, for one, it's how we date fossils. We look at the isotopes, we know how long it takes for an isotope to decay, and then we look at how far it's decayed in that fossil. Now, that is obviously a very oversimplified version of what scientists actually do, but it gives you an idea of how they're used. We can also administer radioactive isotopes to people or animals, and we can trace the atoms because it will attach and fluoresce, and we can trace those atoms through the body. Particularly, this works well in metabolism and diagnostic medicine because you can see those radioactive isotopes upon imaging. But it can also be hazardous to health depending on the cell type and the amount of radiation that you're getting. So medical doctors, they don't use radioactive isotopes on a regular basis, but if they're really needing a diagnostic tool, the advantage to it or the benefit of it could definitely outweigh the possible risk to that. But it's usually it being damaging as a long-term exposure. And with people, if they are using a radioactive isotope for a diagnostic procedure, it's not long-term exposure. This is an example here where the radioactive isotope is labeling cancerous tissues. They're able to see that upon imaging. Again, another review of chemistry. If you don't remember what electron shells or valence electrons or valence shells or electron orbitals are, this would be a good time to go back and review that information again in Chapter 2 of your textbook. And this just kind of reminds you, that electrons can be in different shells, and depending on the shell, depends on their potential energy. And this is just, some typical things that we find in living things that, not all of them, but some of them, find living things, and it just shows like an orbital level of the periodic table. Bonding. Covalent bonds are the sharing of a pair of valence electrons by two atoms. They can either be a double bond or a single bond. Molecules are often held together by covalent bonds. Compounds are molecules of two or more different elements, where we talked about that earlier, so NaCl, because there's two different elements there. A molecule is anything that's more than two, so like O2, but they're not different. So there's two oxygens here, but they're not different. So they're still a molecule because they're being held together. The O and the other O are being held together by a bond, but since there's no other atom besides oxygen, it's not considered a compound. H2 would be in the same category there. But these guys are being held together by a bond, but they're two different elements. Electronegativity, the attraction of a particular atom for the electrons of a covalent bond. The more electronegative, the more pull on electrons. You'll find out that O2 is a very electronegative molecule. A nonpolar covalent bond, two elements have the same electronegativity. A polar covalent bond is when one atom is bonded to a more electronegative atom. Water here is a polar because the oxygen is highly electronegative and the hydrogens are not. We're going to talk a little bit about this when we get to water and its structure and why the makeup of it being polar is very important to some of its chemical properties. And then I already mentioned that oxygen is one of the most electronegative elements. Water is polar because the oxygen has more negative charge and the hydrogens have a more positive charge. And my picture is not showing up. This picture is in your textbook, but it should just show how the electrons are distributed in a couple of different models, and it shows the electron distribution, Lewis dot, if you remember that from chemistry, and then a space filling model, and it just shows several different ones, and that it was a picture just so you could see how bonding happens, but for some reason it didn't pull my picture in. This is water. Again, the oxygens are either on the negative is going to be slightly positive, and that creates a polar. Kind of think polar like a battery, two opposite ends. Ionic bonding is where the more electronegative atom strips an electron from its partner. So here we're stealing electrons. Cations and anions attract each other. A cation has a positive charge and an anion has a negative charge, and so they will attract each other. A charged atom is called an ion. You might recognize ions like potassium or chlorine. I think chloride is how it's typically called when it's an ion. The hydrogen ions, things like that. Compounds formed by ionic bonds are called ionic compounds or salts. All right, sorry. Compounds formed by ionic bonds are called ionic compounds. They're salts. Many drugs are manufactured as salts because they are stable but disassociate easily in water. Remember, you're made out of a lot of water, so anything you take, they want to be able to disassociate very quickly and break down and do whatever it's meant to do. And this is just showing how that ionic bonding happens, where this one is actually kind of like stealing an electron from the other. Weak bonds. Hydrogen bonds are very weak. We'll talk a little about hydrogen bonds when we talk about water. We'll talk about it when we talk about DNA, because DNA is held together by some hydrogen bonds. It's a non-covalent attraction between a hydrogen and an electronegative atom. Van der Waals attractions, they're weak and occur only when atoms and molecules are close together and have changing regions of positive and negative charge that enable molecules to stick to one another. This is why a gecko can walk up a wall and the molecules of the tiny hairs on the feet and the wall molecules form a lot of weak attractions that together can support the weight of the creature. These are Van der Waals attractions are also found in DNA. Molecular shape and function. Shape of a molecule is important to the function in living cells. Biological molecules often bind temporarily, but this can only happen if the shapes are complementary. Lots of drugs, both legal and illegal, mimic different shapes of receptor molecules in the brain and therefore will bind instead. That's why a lot of illegal drugs have mental effects or very, very, very severe biological effects as well as psychological. And that is because something you're normally producing, such as like dopamine, which is kind of like a happy, makes you feel good, serotonin, things that you normally are neurotransmitters that you secrete in the brain. We'll talk a lot more about some of these when we get to the nervous system later in the year. But you produce these, and a lot of times these drugs will actually block those receptors, and so you end up craving that drug, and you need that drug, and you can't get enough of it because it's actually taken the place of something that was there. The important thing to get out of this is that shape matters. In any kind of biological molecule, there is a shape, and it is going to fit specifically with a receptor. That's how your brain knows what receptors to pick up. It's based on their shape. So here's an example with a drug that could just be a legal, you took it in the hospital because you were in a lot of pain, or it could be misused and be illegal, I guess you could say. And it shows you that here's the two structures of natural endorphins and then morphine. And in this, you'll see that this part of the molecule is very similar. Well, this is the reason why morphine works. Okay, the reason it works to block pain is because here's a brain cell, here's the endorphin receptors, and morphine is going to attach to that receptor, and it's going to make you feel like you're not having any pain. The reason why if you get hurt extremely bad, initially you may not feel any pain is because your natural endorphins kick in. But once those subside, we figured out how to manufacture morphine to continue that pain-free feeling. Just another chemistry review. Chemical reactions, making and breaking of chemical bonds, leads to changes in composition of matter. You have reactants giving way to products. Matter is conserved. Atoms are not created nor destroyed, they are simply rearranged. Chemical equilibrium is a relative concentration of products and reactants stop changing. They are not equal, but rather the reactants and products have stabilized to a particular ratio. So the idea here is that's important is when chemicals reach an equilibrium, that does not mean they are not necessarily equal. It just means they are in a ratio that is good for that reaction. Where's my answer? And just a side note that all images I use in any of these PowerPoints are courtesy of our textbook. I pulled them all from there, so if you see any of the image and you really want to go back and look at those or read the caption under it, you can do that. And that was it. And again, if you need to go back and pause at any time to take notes, please do.